viruses. So I love this picture because it really shows you the number of viruses that can exist per cell. So this is the gray part is the cell, and then all of the red dots, each red dot is one virus that is emerging from this cell. Now, do you see how the cell is super lumpy? That is not good for the cell. That generally means death, right? Because for most of these viruses, as they emerge from the cell, they actually pull a part of the membrane off to carry it around like a coat. We'll talk about why they do that later on. But they pull this piece off, and because so much of this membrane is being lost to these emerging viruses, gosh, that cell is essentially being popped, right? It can't survive. Did you have a question? Are you sure? Okay, cool.
No, I just said no, yeah. You said pause. Me, no, just don't worry about it.
All right, I plan to not. So these red ones are all the viruses, and the sheer number, right? The sheer number of viruses that are coming out of this cell are, I would guess, in the hundreds or even thousands, right? And so today we're going to talk about what makes a virus, the fact that a virus is not living. Okay, that's a big deal. Like, wait a minute. I know it makes me sick. Had a cold, had the flu, had chicken pox. So I know the viruses make me sick, but it's not a living thing. So viruses are pathogens. Pathogens are things that make you sick, but it's not a living thing. So we're going to start today by talking about what are the criteria for life? What makes something alive? Because it's important that we see this list and then figure out what's missing from this list with regard to viruses that makes them not living. Okay? Because there are scientists, respected scientists, who believe that viruses are living. Now, for the purposes of your AP biology exam, viruses are not alive. If you want to argue that point with some fancy scientist at some point in your life, by all means do it, but make that happen after May 6th. Okay? So do that later on. Okay? So here's what all living things do or have. They can all reproduce. You're like, well, so far so good, because pretty sure that that's reproduction. Sorry, right? They're reproducing, that's for sure. They have a genetic code. Actually, viruses have that too. All viruses have either DNA or RNA inside of them. Respond and adapt. Viruses actually don't do that. Use and obtain energy. Depending on your perspective, they might do that one. Evolve. Also, depending on your perspective, they might do that. Grow and develop. They absolutely do not grow and develop. The last two are the kickers. Organization, meaning they're made up of at least one cell. Maintain dynamic homeostasis. To be a cell, you have to be surrounded by what? Plasma membrane, right? Viruses are not. Viruses are not surrounded by a true membrane. Remember the membrane, the feature of a membrane that makes it so important is that it is selectively permeable. We spent a lot of time talking about that back in Unit 2, was the selective permeability of that membrane is what allows those cells to maintain dynamic homeostasis. So viruses that lack a cell membrane, meaning they don't have a cell, also are not capable of this dynamic homeostasis. So these are the features that really cause viruses to not be living. Because the cell theory is that all living things are made up of cells. And a virus is not. Okay, it's not made up of cells. So then, well, okay, so what is it? Viruses do not have, this is what I was saying before, they do not have that true membrane. They have no organelles. And as a result, they cannot carry out metabolism unless they are inside of another cell. They have to be inside of a cell in order to carry out metabolism. They are generally considered not alive, but exist in this very shady region between living and chemicals. Viruses alone are biologically inert. They're not cells, they're infectious particles. For reasons that are not clear to me, I put the same slide in twice. Okay, so virus isn't. So what is it? Well, chemicals. It's a macromolecules. Okay? A viral genome is DNA and RNA. One or the other. DNA could be single-stranded or double-stranded. It could be RNA or it could be DNA. So a virus has a nucleic acid. It has genetic material. Conveniently, we call viruses RNA viruses or DNA viruses depending on the nucleic material. I like it when the words make sense. And in this case, they do. That genetic material, that nucleic acid, is surrounded by protein. That's all a virus is. So when I told you it lives in this shady area between living and chemicals, really all a virus is a nucleic acid, RNA or DNA, surrounded by a protein coat. Well, that's a little bit tricky, because how do you make a protein? I mean, what organelle is necessary? Ribosomes are absolutely necessary in order to make a protein. So, a virus is surrounded by protein. Protein is made with ribosomes, but does a virus have ribosomes? Nope. So now we're into this whole bit of trickery, right? So if the virus is DNA or RNA surrounded by protein, but the virus can't make its own protein, what does it have to do? Well, it has to hijack the ribosomes of another organism. Okay? So here's examples of viruses. What's that? It looks like a spider, I know, right? We use... Ah! Yes, it looks like a spider. Right? Or something that would be in one of those mummy movies that go like, exist in millions and they're scattered across the floor, right? I feel confident. Yes! We should all fear this. The good news is that it only infects bacteria. So this is a bacteriophage virus. That's what it is. So phage means eater. So this is a bacteria eater. So if you're a bacteria, this fear would be well-founded in reality. Okay. Instead, we should fear this one. This is the flu. Okay, this is high on the list of most deadly viruses to humans on the planet. We don't think much about the flu because most of us in developed countries survive the experience. But worldwide, influenza is extraordinarily dangerous. Kills a lot of people. But this is what viruses are. Viruses are nucleic acid surrounded by protein. Okay, nucleic acid surrounded by protein. And so the protein is something we call a capsid. This is saying here. The protein shell that encloses that genome is called a capsid. The capsids themselves are made up of individual protein subunits called capsomeres, and they come together in a variety of different shapes. And so this is called the tobacco mosaic virus. This is a plant virus. Okay, the adenovirus. Okay, this is one that can infect humans. See, it's got little spikes coming off of it. You can actually see the spikes down here. This is the flu virus, and this is a bacteriophage. Some structures, we're going to zoom in on this one real quick. Some viruses have these things called accessory structures that help them infect their host. For example, this membranous envelope. Well, remember that viruses make me sick. And I have a whole system dedicated to not getting sick, right? Called my immune system. And that system's job is to look for nasty things and assault them. So it's one job, right? It's one task. So the virus wants to hide from my immune system. How do you hide from my immune system? Well, you look like you belong there, right? Okay, it's kind of like when you want to walk into the movie theater with the Starbucks. How do you get away with that? You walk like you belong there. That's how it works, right? If you walk in trying to be all sneaky, they peg you and they're like, what are you hiding? You walk like you belong there, no problem. That's how that works, right? That's how you get Starbucks and to go see Star Wars. So you have to look like you belong there. How do you look like you belong there? Well, you surround yourself in a piece of membrane from the host cell. You're like, what? I belong here. Leave me alone. So we call this thing the membranous envelope. The membranous envelope. And it is actually a chunk of cell membrane, but the virus didn't produce it. Where did the virus get it? the last cell it infected, when it bubbled off, we're going to go back, when this guy's bubbling off, they actually take a chunk with them, almost like a vesicle, right? It's almost like a vesicle that's being released. But as that virus emerges, it's like, I'm just going to take a little bit of this. We're just going to pretend like I belong here so that the immune system doesn't see me and go, ah, I think not. And then assaults the virus, and that's all there is to it, right? So how does the membranous envelope help? Well, it consists of membranes, along with all the stuff that was in the membranes, proteins and glycolipids, from the host cell. And then there's also proteins and glycoproteins from the virus. And all of those things together help the virus not only hide from the immune system, but to also be granted entry into uninfected cells of the host. Some of the most complex capsids are actually those that infect bacteria. Right? This is much more complex than the other things that we were looking at. Okay? And that is your bacteriophage or bacteria eater. The most complex viruses are actually the ones that infect bacteria. So we talked before, viruses don't have enzymes, they don't have ribosomes, they don't have all of the parts that they need in order to make proteins, in order to be self-sufficient. Okay? And so if you think back to when we talked about molecular genetics and protein synthesis, we talked about, okay, so first, at least in eukaryote, we have transcription, and then we have translation, and then with transcription, we have this RNA processing, and then the mRNA left, and then we had all of these different moving parts, like ATP and GTP and ribosomes, that all came together to form this complex. It was very complicated. Viruses don't have any of that. They don't have any of the stuff that they need in order to produce these proteins. So as a result, we call them obligate intracellular parasites. They are required to be inside of a cell, and they're bleaching the stuff out in order to survive. A virus just on a table can do nothing. Okay? But as soon as that virus infects a host, then the virus can do things. Unlike a cell can maintain dynamic homeostasis all by itself. A cell can do things by itself. Okay? A virus cannot. Viruses can only replicate within a host cell. Really, all that a virus is a set of genes that moves from cell to cell. That's it. How does it move? Proteins. Not all viruses can infect all cells. When I was young, I remember asking my mom at one point, Mom, how come the dog never gets sick? I get a cold. This is a fair question, right? I mean, I'm like 8, and it makes no sense. We have pet dogs. I get sick. I get sick, and I give it to my brother. My brother gives it to my sister. The whole house is sick, right? We all go through this. How come the dog never got the cold? My mom, tired of my questions, having 3 children, and really just likes to end things as quickly as she can, says, God did it. That's just how it is. Because really, when you're 8, how do you come back with that, right? I got older and was like, okay, I feel like there must be more to it than that. And there it is. Why does the dog not get my cold? Again, a legitimate question. The answer is the same reason as how come when I get a cold, it's always up here? How come I don't get a cold in my foot? Can you imagine you walking down the hall? Oh God, not the right one. Can you imagine a situation where you got a cold in your foot? This is ridiculous, right? Okay, but it is actually the same question as how come the dog doesn't get the cold? Right? Why is it always up here? And it's because viruses are very particular to only being able to infect not only just one species, but one type of tissue within the species. Okay? We call that a host range. So viruses are obligate intracellular parasites. So we just said that. But which cells it can actually invade is a very small range for most viruses. So, the flu, mucous membranes, HIV, white blood cells, okay. Polio, nerve cells. Okay. So when you think about viruses, chickenpox, skin cells, right? When you think about viruses, we don't tend to think about, well, what tissue is it actually infecting? But that's a part of the plan here, right? You don't get chickenpox in your bones. You get chickenpox in your skin and all over your skin, anywhere you have skin, right? Okay? But nowhere else. So we call that, again, a host range. Now, some viruses can jump species to species. Do you remember swine flu? Several years ago now, swine flu. One of the issues with swine flu is that epidemiologists, people who study viruses and other things that make you sick, there was a virus that jumped from one species to the next. And that was cause for huge concern. Because that makes it much more difficult to control. We can control things through quarantine, things that impact humans, right? And we're like, oh, you're sick. We're going to make you hang out in here in the bubble room, right? If it's Ebola, right? Here you are in the bubble room. But when it starts to infect things like birds, can you tell me something that infects birds and humans? It's a problem in Colorado, which there is a bird flu, but that's not the one I'm digging for right now. This is real common. You may have had it. West Nile. West Nile is a virus. West Nile virus can go from birds to humans. You're like, okay, but how do I get West Nile from a bird? It sneezes on me. Oh, man. A mosquito. It poops on you. No. A mosquito. A mosquito bites a bird that has West Nile. The same mosquito bites you that has West Nile. A mosquito in this case is called a vector. And that mosquito moves things from place to place, right? West Nile can be passed between multiple species. Do you know, I mean, you guys hear about West Nile in the news pretty regularly. I remember you guys don't remember the late 90s, but I sure do. And when West Nile was first an issue in North America, it was probably on the news every night. Every time there was a new case, another case of West Nile. Don't go outside without bug spray. You know, long sleeves, long pants. You know, protect your neck, the whole 9 yards. And it's because when these viruses start being able to jump from species to species, that's when it becomes an awful lot more difficult for us to control them, right? For us to manage them as a risk. Okay. So most viruses, very narrow range, right? Like HIV, like a cold, like the flu, only infect one species and only one type of tissue within that species. But there are a handful that can jump. And those are the ones that you hear about on the news. Okay. Those are the ones that are the big ones. So how do viruses figure out what to infect? Well, it's a lock and key fit between proteins, just like everything else is. Right? That complementary structure of proteins on that virus, the exterior portion of that virus, to the cell that it's trying to infect. Okay? Once we get that lock and key fit, what happens typically is the cell goes, oh, come on in, buddy. They let them in, and it's like a Trojan horse. Comes in, and out they explode. Trouble begins. The viral infection begins when the virus binds to the host cell, and the viral genome makes its way inside. So this is how you get the flu. Okay? So it's like it's kind of it's a hit and destroy, and then kill, and then run. There's death involved. So we have that membrane, right? And that membrane came from the last cell that was infected. And inside that membrane is the nucleic acid and the protein. So this flu virus has evaded your immune system, has hidden using this membranous envelope, and then been allowed in. And it comes in, the cell engulfed it via endocytosis. We'll learn about that in Unit 2. And as soon as it comes in, it releases all of the stuff that's inside. Okay. The viral RNA goes into the nucleus where RNA polymerase replicates it. Did the virus come with its own RNA polymerase? So where does RNA polymerase live in my cells? Where do I make RNA? In my nucleus, right? Because when I go through transcription, let's put this on the board. When I go through transcription, where does that happen? what enzyme am I using? RNA polymerase. So if the virus wants to make copies of its RNA molecule, we need RNA polymerase to do that because RNA polymerase is the enzyme that makes the polymer RNA. That means. So we go into our nucleus. So here it is. Went to the nucleus and said, look at all that RNA polymerase. Can you just make a bunch of copies of my RNA? The viral mRNA, meanwhile, out here, so we take this, we release it here, the viral messenger RNA is then used to make the viral proteins. So we took the little bit of RNA we had here, it goes into the nucleus makes lots and lots of copies of it, then leaves. Out here, we hijack all of the proteins that are needed and the ribosomes that are needed in order to make proteins. So now the cell, instead of doing whatever that healthy cell is supposed to be doing, now the cell is making viral RNA, viral proteins, they reassemble into this, and then they bud off. And this diagram's only showing you 3 babies, but I showed you that diagram at the beginning where there's hundreds. And look what happens as he's leaving. It can steal some of that membrane. Okay? And that's the envelope that it uses to hide the immune system in the forward. That viral DNA, the nucleic acid, can be taken into the host cell by a variety of different means. Remember, the bacteriophages inject it. Could also be endocytosis, where literally the cell's like, oh, sure, come on in. Fusion of the viral envelope with the host plasma membrane, okay, similar to endocytosis. But once inside, the proteins that are encoded for by that viral genome literally commandeer the host. They hijack that cell, reprogramming the cell so that instead of doing whatever its job is to keep you healthy, it copies the viral nucleic acids and manufactures those viral proteins, just like we saw in the diagram. The host provides the nucleotides, the enzymes, the ribosomes, the tRNAs, the amino acids, the ATP. Put a star next to this. All these things come from the host cell. The DNA, or the nucleic acid, is the only thing the virus contributes. Everything else is hijacked from the host cell, that whole list of stuff. And that means that the cell can't do whatever it needs to do in order to stay healthy in the first place. So after the viral nucleic acid molecules and those capsomeres are produced, they spontaneously self-assemble into these new viruses, and the cycle ends with the exit of all of these new viruses, hundreds or even thousands of them. This process usually damages or destroys the host. And so here's the diagram all in one piece. Okay, so this is the same diagram as this one, only all together here. So the virus goes in, it breaks apart, we copy everything we need to copy, it all self-assembles, and everything leaves. So that is a very general virus cycle. I don't want to call it a life cycle because it's not living, but it's a replication cycle for a virus. The cellular damage, okay, those popped cells, the cells that are no longer able to do what they're supposed to do, cause many of the symptoms that are associated with viral infections. Okay, so what makes you feel gross isn't the virus, it's your body's response to the virus that makes you feel awful. So the viral babies, the progeny that leave, then go out to infect other cells. Lots of variations to this typical reproductive cycle. We're going to look at 2. We're going to look at viruses that infect bacteria, and then we're going to use retroviruses like HIV to understand the connection to genetic engineering. First, bacteriophages. These are the spider-looking ones. Okay. 2 different mechanisms by which viruses can replicate. The lytic cycle and the lysogenic cycle. What does it mean to lyse? To cut, to pop, yeah, to lyse. Yeah, it's a little crazy in here, right? So, to lyse means to pop. So we have these 2 cycles. We have the lytic cycle and the lysogenic cycle. And the stage that culminates in the death of the host cell is the lytic cycle. Because at the end, this happens to the cell. Okay, it pops. Done. Okay, lyses. A phage, so right now we're talking specifically about viruses that infect bacteria. A phage that only replicates by this lytic cycle is called a virulent very similar to this one. Okay, where it goes in, it hijacks the cell, and it produces a whole bunch more. Okay. So what you're seeing, we're looking at bacteria, which is now why we have one circular chromosome, no nucleus. Okay. But here's our spider dude. He poops his DNA in here and hijacks the enzyme, degrades all of the functional post-DNA, uses the enzymes that are floating around in the cytosol to replicate its own DNA and protein. These things self-assemble, and look at the puzzle that we have here. Fancy. They self-assemble and literally explode out of the cell. And then these guys float around and do it again. So you might be thinking, okay, this seems pretty aggressive, and it always ends in death. And the difference between this and the flu is that this is the whole organism. So getting a viral infection if you're a bacteria means you're dead. Right? Where I get the cold, and that tends to work out in the long run. I might not be real happy about it, but death doesn't usually follow. So you may be thinking, how do we still have bacteria if one viral infection means the bacteria is dead, and look at all of those new viruses? Well, bacteria have been around a long time. So they have a way to fight back. Right? They have a way to fight back. Bacteria have their own defenses. Natural selection will favor bacterial mutants with receptors that are no longer recognized by a particular type of phage. So the whole thing begins here, that the phage recognizes the bacteria as a host. So natural selection is going to favor the bacteria where there is not a virus that sees it as a host. Those will live longer, reproduce more. When phage DNA does go in, the DNA is identified as foreign and cut up by cellular enzymes. So in step 2, I told you this guy goes in, hijacks the machinery in here, and digests the DNA. Well, some bacteria have wisened up to this and have something called a restriction enzyme that knows what foreign DNA looks like, and when it comes into a cell, it's like the pirate, remember? Or the cop in the video we saw with the purple flowers. Do you remember? Remember the cop in the castle and the nucleus? Right? So sometimes this gets inserted, and the cop that's inside goes, right? And destroys this before it can destroy this. The name of the cop is called a restriction enzyme. Restriction enzymes. The bacteria's own DNA is methylated. We know what methylated DNA does. What does methylated DNA do? Condenses. Right? Is methylated in a way that prevents those restriction enzymes, the COP, from destroying its own DNA. So the COP is looking for DNA that is not methylated, and is like, aha, you don't belong here, and I'm going to destroy you. Okay? We use restriction enzymes to cut up DNA in biotechnology. We'll deal with them a lot in these next few lectures. Lots of phages manage to coexist with bacteria in a state of lysogeny. So I told you a second ago, so this was our lytic cycle. Okay. A lytic cycle, we can replicate by the lytic cycle or the lysogenic cycle. The lytic cycle is where at the end it explodes. The lysogenic cycle allows the DNA, the nucleic acid of that virus, to be replicated in the genome without destroying the host. I'm going to go forward a slide and then we'll go backwards a second. So this is what we just talked about. We just talked about the lytic cycle. Comes in, gets inserted. We skipped a stage where this gets digested, and then we make these, and they all pop, right? So we were just talking about this lytic cycle. Okay, that's what we were just saying. But we have this other cycle called the lysogenic cycle. This is sneaky, because the phage will insert the bacteria, or insert the DNA in the bacteria. Here it is. But instead of hijacking the cell and killing it, this phage DNA gets actually inserted into the DNA of the bacteria. In this case, we call it prophage. So it's inserted, but then as this cell replicates by binary fission, remember that's how prokaryotes replicate by binary fission, that phage DNA is replicated right along with the host DNA. So this is what we get. So this cycle, okay, can go on and on and on and on and on indefinitely. And in this case, the bacteria doesn't die. It's just the viral DNA is hiding inside. But then some kind of an environmental cue, a chemical signal, will signal the prophage DNA to literally pull out like this and cause this stage to happen. So this can go on for years, okay, and allow the bacteria to replicate. But ultimately, we can get back to this stage. Over here. Let's go back and make sure I said everything I needed to say. Oops. Phages that are capable of doing both cycles, lysogenic and lytic cycle, we call them temperate phages. But during that lysogenic cycle, the DNA is incorporated into the bacterial chromosome, and it's mostly silent, but it persists there through all the replications. And when the phage DNA gets passed from daughter cell to daughter cell, it can be activated at any point. Question or stretch?
Stretch.
Question.
Are all lytic,
Lytic?
are all lytic, lysogenic? Or are they, are all lysogenic all?
Yes, so the lysogenic site, you would never have this happening by itself because you would never actually produce a virus at the end. So if you do this, then it can revert over here. Okay? But you can have the lytic cycle by itself. Okay? You can have the lytic cycle by itself. And remember, guys, this is just for bacteria. This doesn't happen in humans. Okay? We have something kind of similar happen with HIV that we're going to talk about here in just a second. But this whole lytic lysogenic thing, this is just bacteriophages. Okay? Just bacteriophages. So we're going to look at the virus life cycle exception number 2, retroviruses. Retroviruses are viruses that work backwards. A little bit of background first. So this is normal. So we're going to talk about normal here again one more time. An animal virus has that outer envelope that allows it to hide from your immune system. And it uses that to enter the host cell. Once it gets in, then we take the viral genome and we replicate it, and we make proteins out of it. And then all of that reassembles and it leaves with a chunk of the membrane as well. Right? That's what we were saying before. So what does a retrovirus do? Well, actually, I'm going to write on the board first. So this is what we have. We learned this last time, or last semester. We called this the central dogma of biology. And I'm going to write smaller now, so this was transcription. What was this one? Translation. See, look, you remember something. Right? This is what we have. And of course, sometimes we get stuck here and we have ribozymes, or, you know, and sometimes we have different, just a polypeptide. We talked about all those exceptions back in Unit 2. But we said this is the central dogma of biology. This is how the information goes. Well, retroviruses, retro meaning backwards, can do this. And they use a fancy enzyme for this called reverse, get this, transcriptase. The enzyme that does transcription backwards. Okay? Well, transcription normally takes DNA and turns it into RNA. Well, reverse transcriptase can take RNA and turn it into DNA. Why is this useful for a virus? Well, there are 3 types of single-stranded RNA genomes that we can find in animal virus. We can have a viral genome that can directly serve as messenger RNA and can be translated directly into a viral protein. We can have an RNA genome that serves as the template for mRNA synthesis, or we can have a retrovirus. And the retrovirus are those things that are equipped. Who are you looking for?
Hi, everyone.
Hi. Thank you. The retroviruses are the ones that are equipped with an enzyme, that reverse transcriptase, which transcribes the RNA template into DNA And that gives us a backwards information flow. So, an example of a retrovirus is a, thank you, see you later. So in this case, so here's RNA, or here's our retrovirus that's carrying RNA. It comes in, here's your viral RNA with your reverse transcriptase. It makes this RNA-DNA hybrid. Once we have one strand of DNA, your cell already has the enzymes that it needs in order to make the other strand. But then we have this chunk of DNA that actually goes into the nucleus and inserts itself into the chromosomes. So this is what HIV does. And this is why HIV can have such a long period of time between getting infected and you showing symptoms. Because it goes in and it just literally hides on the inside. And rather than all at once producing a whole bunch of viruses that explode from a cell and kill a cell, it very slowly replicates just through transcription and translation, just like we learned in Unit 4, produces the mRNA, codes for the proteins, and it escapes. A little bit at a time so as to not kill the white blood cell. Slowly, 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 slowly. So an individual that has HIV, their doctors are looking for a reduction in white blood cell count in order to see just how sick you are. Because the longer the virus has time to do this, the lower and lower and lower over time your white blood cell count gets. This new virus then goes out and infects another white blood cell, and the process continues. At this point, there are a lot of different drugs that can be used to slow this process down, and having HIV is not nearly the death sentence it was when I was growing up, but still something that you have forever. Right? There is no cure to any of this. Here's why this matters to us in the world of biotechnology. Imagine I am a physician, and I am trying to help people with diabetes. And so I want bacteria to produce insulin. This is what I want bacteria to do. Okay, so now let's do a little bit of review real quick. This is actually how insulin is produced. Okay, bacteria. Do they do this? DNA becomes RNA, RNA becomes protein. Is that what they do? Yeah, right? This is the central dogma of biology, right? Yes, absolutely they do this. Nucleus or no nucleus in my bacteria? No nucleus. So, is there a time or spatial separation between transcription and translation? No. Does it all happen at the same time? Yes. So, does mRNA processing happen in a bacteria? No. So, are there spliceosomes to remove introns from the messenger RNA in a prokaryote? No, there are no spliceosomes. Okay. So far, we're good. Except, we have a problem. Because if I go looking in my DNA for the gene for insulin that I'm going to try to put in an E. coli, so this here is the human gene for insulin. Intron, exon, intron, exon, intron, exon. Let's say. Well, in me, this whole thing is transcribed, is it not? And then we have mRNA processing, and what happens to my introns? They get cut out, right? So then, before that gene leaves my nucleus, all I have is a bunch of exons, right? And then this is my mRNA, and where does it go? To the ribosomes, right? Like this is, we learned this, we remember it, at least vaguely, I hope. Right? So this goes out, okay, and then my ribosomes produce insulin, right? I can't put this gene in a bacteria, because what will the end result be? This, exactly. It's going to translate my introns and my exons into a polypeptide, and will the result be insulin? No. HIV taught us how to take this, our messenger RNA, and turn it into DNA. Because we have this enzyme called reverse transcriptase, it's the green dot. So how do we make insulin? How did we make that gene originally? We took the mRNA, we treated it with reverse transcriptase, we got the DNA out of it. Where do I put that gene? Into the E. coli. And now, when the E. coli goes through the process of transcription and translation, it will make insulin. Whereas if we didn't have reverse transcriptase, would it have made insulin? No, it would have made this thing with all the introns and the exons. So when we talk about genetic engineering, when we talk about producing a protein, a eukaryotic protein, but producing it in a prokaryote, we have to get out the introns. How do you get out the introns? Well, you take the RNA, you treat it with reverse transcriptase, and that's how you get rid of the stuff that you don't need. As devastating as HIV is and was, it taught us most of what we're now able to do with biotechnology. That one enzyme right there, reverse transcriptase, allowed us to backwards engineer prokaryotes to produce eukaryotic proteins by removing those introns using reverse transcriptase. It's pretty wild. And that's why we learn about viruses before we do genetic engineering. Okay, so why do viruses suck so much? Well, they make us sick. A viral infection can produce symptoms by a number of different routes. The viruses may damage or kill cells by causing the release of hydrolytic enzymes from lysosomes. That's bad. Remember, the hydrolytic enzymes are the things that break down polymers and monomers. Right? So a virus can actually allow all those things to flow freely in the cell and break down all of your macromolecules. That's troubling. It can cause infected cells to produce toxins that lead to disease, or have molecular components that are toxic themselves, such as those envelope proteins. But how much damage a virus causes depends on the ability of the infected tissue to regenerate. So why don't I die from a cold? Well, because all the cells that die 2 or 3 weeks later have all been regenerated, and now I'm fine again. But the problem is with things like polio. With polio infects your nerve cells. Nerve cells don't replicate. So damage done by the polio virus is lifelong. You never recover from that because your nerve cells can't regenerate. So how much damage a virus itself does depends on the ability of those cells to regenerate. Vaccines are without question the best defense against viruses. Vaccines are harmless variants of pathogens. They stimulate your immune system to mount defenses against the harmful pathogen itself. When we get to Unit 7, we'll zoom in on that and talk about specifically how does a vaccine achieve that. So while prevention of a viral infection is readily available, cures do not exist. You cannot be cured from a viral infection. You only can wait for your body to solve the problem itself. Antibiotics are only for bacterial infections. Okay? And so if you have a cold, you go to the doctor, they can give you stuff to make you feel better in the short term, but ultimately you won't be healthy until your body solves the problem. That's really what it comes down to. Viruses that suddenly come onto the scene are often referred to as emerging viruses, viruses that didn't exist before and then suddenly do. HIV was an emerging virus in the late 80s. West Nile was emerging in the late 90s. Sometimes we call this an epidemic, okay, or a general outbreak. How does this happen? Like, where do they come from to cause these outbreaks? Well, it could be that an existing virus mutates. Okay, so because the DNA that's in a virus doesn't have to actually do anything, there is no risk to mutation, right? They remember as viruses are moving from bacteria to bacteria or cell to cell, they literally just pick up nucleic acid and leave. Well, what they pick up is kind of random. They just collect it from the cytosol and take off. So there's mutations all the time in viruses. That's how come you get a cold every year, because over the course of that year, those colds have mutated. A virus could be disseminated widely when it was previously isolated. That's what we see with Ebola in developing countries in Africa or undeveloped countries in Africa. Very isolated. We try to keep it isolated, but if it gets out, that's when we have massive concerns. Third, a virus is able to spread from other animals to humans. And this is the one that becomes most scary, because we can't control animal movement as easily as we control human movement. And so this is very dangerous. Of course, plants are susceptible to viruses as well. Okay? There are more than 2,000 viral diseases for plants. They have the same basic structure and mode of replications as we were looking at before. This eggplant, this is a healthy eggplant, and these are unhealthy eggplants. this eggplant is infected with the tomato bushy stump virus, which is hilarious to me because that's not a tomato. But whatever. Okay? Apparently it also affects eggplant. Viral diseases in plants can spread by what we call horizontal transmission or vertical transmission. Horizontal transmission just means that a plant is infected from an external source. Okay, so it's floating around the air and it gets into the plant. Vertical transmission means the plant inherited it from a parent, which is not uncommon either. But once a virus gets in and begins replicating, the viral genomes are associated with proteins can spread by means of plasmodesmata. Remember, plasmodesmata in plants were similar to gap junctions in animals, and that was just the connection between two cells that allowed stuff to flow freely between them. Plants have those, a lot of plasmodesmata. So once one cell is infected in a plant, it just spreads through the whole plant very, very quickly. Scientists have not devised cures for most viral plant diseases, mostly because, well, we can't even cure them in humans, so who's to think we could cure them in plants? And second, there's really not a lot of financial investment in figuring it out, because if plants get viruses, you just chop them down and plant a new one. So there's just not the research money on that one either. So that's your down and dirty about viruses. Let me pause this.